This method can help to answer key questions in the centriole field, such as localizing protein to a specific region of the centriole. The main advantage of these techniques is that we provide a means to concentrate centrioles to get them in multiple orientations.
Though this method can provide insights into Chlamydomonas centriole biology, it can also be applied to other systems, such as isolated human and paramecium centrioles.
Demonstrating the procedure will be PhD student Nikolai Klena, another PhD student Maeva Guennec, and a postdoc in the laboratory, Davide Gambarotto.
To begin this procedure, first prepare all media and culture the C. reinhardtii cells as outlined in the text protocol. Transfer the prepared cells to 50 milliliter conical tubes and centrifuge them at 600 times G for 10 minutes. Wash the pellet once with 50 milliliters of 1x PBS and spin the cells again at 600 times G for 10 minutes. Use a pipette to resuspend the pellet in 100 milliliters of room temperature deflagellation buffer. Place the beaker on a magnetic stirrer and slowly add 0.5 molar acetic acid to a final pH of 4.5 to 4.7. Let this mixture incubate at room temperature for 2 minutes. After this, slowly add drops of 1 normal potassium hydroxide to restore the pH to 7.0. Transfer the cells to 50 milliliter tubes and centrifuge them at 600 times G for 10 minutes to remove any detached flagella. Discard the supernatant and store the pellet on ice until ready to wash. When ready, wash the pellet twice using 50 milliliters.